tissue engineering. It's an emerging field of biomedical science that holds great promise for helping warfighters recover from injuries or disease, and one poised for a major expansion. By one estimate, the global tissue engineering market will reach $11.5 billion in value by the year 2022, driven in part by advancements in 3D bioprinting and non-embryonic proliferative stem cell research. Hi, I'm HDIAC subject matter expert Joel Hewett, here to discuss how tissue engineering, or the creation of functional replacement tissue for clinical use, can help the Department of Defense treat debilitating diseases and repair or replace damaged tissue and damaged organs. The National Institutes of Health define tissue engineering as the practice of combining scaffolds, cells, and biologically active molecules into functional tissues. This approach translates into a wide array of clinical applications. For example, researchers have grown acellular blood vessels for use as vascular grafts in humans, and produced articular cartilage tissue around scaffolds to repair injured joints. DoD has shown significant interest in advancing tissue engineering technologies and other components of regenerative medicine. In fiscal year 2019, for the first time, the DoD's Clinical and Rehabilitative Medicine Research Program convened a special programmatic award panel for regenerative medicine, drawing from expertise at DoD entities like the Defense Health Agency, NIH, and the VA's Extremity Trauma and Amputation Center of Excellence. Tissue engineering is rapidly advancing, but one of the major challenges that the field faces is figuring out how to map cell growth processes in 3 dimensions. One of the leaders in this field is Dr. Hwan Lee of Purdue University, who next explains the approach his lab takes when it comes to regenerative medicine.
Hello, my name is Hwan Lee, and I'm an assistant professor in biomedical and mechanical engineering at Purdue University. So my lab focuses on the development of mechanically flexible and stretchable bioelectronics and sensors that can be upscaled to the human body integrated healthcare systems and downscaled to the cellular and tissue engineering applications at their length scale. At Purdue University, we developed a new engineering concept of tissue scaffold where biological cells can grow into functional tissues like artificial skins, cartilage, and bones in order to improve or replace damaged tissues. In this field, our breakthrough is the electronic tissue scaffold system that contains tiny functional sensors embedded inside of the biocompatible polymer materials and therefore provide the ability to detect the cellular behaviors and tissue functions in a real-time fashion. This real-time monitoring of physiological or electrophysiological biosignals provide important insight into human cells and tissues to better understand disease or wound healing process, which is the prerequisite for developing target therapies for specific pathologies and individualized patients.
Recording cellular behaviors and functions in real time and at high spatial and temporal resolution provides researchers the ability to model diseases and understand the underlying biophysics of different tissue structure types. However, the liquid culture mediums often used in tissue engineering don't mix well with electronic instruments. Bridging this gap is the focus of Dr. Lee's most recent work.
However, the long-term reliable recording of the cellular functions has been challenged by the conditions required for culturing biological cells, like maintaining a cell medium culture solutions at 37 degrees C with carbon dioxide. And this is entirely unfavorable to electronic instrument settings. So we tackle this problem by developing a unique and sponge-like materials with lava-like properties that are so buoyant such that the entire tissue scaffold system float on the surface of the cell medium where all the electronic components are placed in the air, while the cells reside and grow on the bottom of the tissue scaffold, which is submerged in the cell medium solution. So this setting provides a hospitable environment for both biological cells and electrical measurement settings, allowing us to monitor the cellular functions over a long period of time, actually more than several weeks, with high fidelity.
This scaffold system allows researchers to monitor tissue development via cell substrate impedance, as well as electrophysiological signals. This breakthrough is likely to find widespread use in the field, as Dr. Lee explains.
So this creates a research platform for analyzing the cell biology in a precisely controlled environment. For example, to understand how well the cells are adhering to one another, how successfully they are proliferating, how individual cells respond to external electrical signals or drug molecules of interest, and how they interact with their environment and organize into tissues and organisms. This will eventually allow us to manipulate these cellular behaviors and processes to replace damaged tissues with the newly created one. There is also future potential for using this electronic scaffold platform for long-term monitoring of tissue function during and after transplant into a patient. It might be also possible to construct naturally dissolvable form of the electronic scaffold, tissue scaffold system such that its materials degrade in the body in a harmless manner following the implantation and post-implant monitoring. So this can eliminate the unnecessary need for post-surgical extraction. So the future possibility is unlimited.
Dr. Lee's lab is already testing his scaffold device in collaboration with other researchers at Purdue University in hopes of advancing stem cell therapies for regenerative treatments. For more information on this topic, click on the link in the description below to see our journal article on tissue engineering for a better tomorrow. Thanks for joining us, and don't forget to follow us on social media.